Welcome everybody. I'm Bettina Winkler from Visiopharm and I would like to welcome you to our virtual Research Rebellion tour. Today we're kicking off the series with Dr. Namrata Singh, who is Technical Director of the Dana-Farber Cancer Institute Core Pathology Lab and also holds a concurrent research position at Harvard Medical School. She has nearly 3 decades of experience spanning cancer research, spatial biology, and histological analysis, bridging the gap between technology and translational research. Namrata will talk about how AI and spatial proteomics enable high-plex protein-level mapping of cells with intact tissue architecture, providing insights into both expression and localization patterns, unlocking the tissue microenvironment for next-gen cancer drugs. She will answer your questions after the talk, so please add any questions to the chat. With that, let's get started.
Hello, everyone. My name is Namrata Singh. I'm a technical director at Dana-Farber Cancer Institute at Pathology Core Lab. It is a privilege to discuss the convergence of two or more transformative frontiers in modern oncology. For decades, our understanding of tumor microenvironment has been limited to viewing it as a disorganized collection of cells. However, we now recognize that in the context of oncology and therapeutic resistance, location is paramount. Much as an architect must grasp the intricate structural integrity of a building to refine its design, we must decipher the complex architecture of tumor to effectively dismantle it. Today, I'm happy to present how we are leveraging artificial intelligence to bridge this gap, transforming raw data into spatial intelligence to catalyze the discovery of next-generation cancer therapeutics. The topics I'll be covering in my talk today are tumor microenvironment and spatial proteomics, why spatial proteomics and AI matters for oncology. I'll be sharing some case studies and the workflows for drug target ID and biomarker discovery, and some challenges associated with these methods. To truly unlock the complexity of tumor microenvironment, we have developed a seamless end-to-end workflow that integrates high-plex imaging with advanced computational analysis. It begins with spatial omics phase, where we utilize high-plex immunofluorescence to capture the precise location of dozens of biomarkers simultaneously within a single tissue section. This raw spatial data is then processed through our AI-powered analysis engine, which performs automated cell segmentation, phenotyping, and neighborhood analysis to decode the social network of the tumor. Finally, this leads us to actionable insights, where we translate these spatial patterns into quantifiable data that can predict drug response, identify new therapeutic targets, and ultimately accelerates the delivery of next-generation cancer treatment. Tumor microenvironment. Why do we need to know about tumor microenvironment? Cancer is currently the second most common cause of death in the United States and is likely to become the most common in the future. Tumor microenvironment is an ecosystem of immune cells, fibroblasts, vasculature, signaling molecules, and extracellular matrix that surrounds and interacts with tumor cells. It actively shapes tumor growth, immune evasions, metastasis, and therapy resistance, making it a critical determinant of cancer progression. Because of this, the TME is both a major barrier and a promising target for effective cancer therapies. Spatial proteomics technologies have rapidly advanced and now key to Atlas-scale projects, enabling detailed mapping of tissue proteomes and enhancing our understanding of health and disease. Spatial proteomics encompasses a diverse array of technologies that vary in scale, resolution, sensitivity, offering researchers and clinicians a broad range of tools to investigate the spatially resolved proteome. The depth and resolution of spatial proteomics, and in particular deep visual proteomics, makes these technologies incredibly valuable in biomedical research. Spatial biology is revolutionizing biomedical research by emphasizing 3D relationships and arrangements of biological molecules within a tissue, exploring cell phenotypes, correlated locations, and cell-cell interactions in the tissue microenvironment can help understand biological processes and disease pathogenesis. Integration of technology and biology. Why now? Availability of high-dimensional dataset, including multiomics, high-performance computing, decline in the price of massively parallelized computing power, innovative machine and deep learning algorithms. These tools with biological data, researchers can better understand disease, identify new drug targets, and personalize medical treatments. This convergence is accelerating discoveries and enabling more precise and predictive biology. Now I'll be sharing some case studies that I worked in my lab with you all. I have nothing to disclose with respect to this presentation. This slide illustrates the evolution of pathology at DFCI core pathology lab across 3 generations. The first generation relied on traditional glass slide diagnosis, where pathologists manually examined samples under microscopes. The second generation introduced whole slide imaging, allowing slides to be digitized and viewed on computers, improving storage and sharing of data. Third generation incorporated Phenocycler Fusion, a COA platform, an AI-based algorithm, Visiopharm. This AI-based algorithms in pathology has assisted in analyzing digital images and identifying disease patterns. Overall, this progression shows how technology is transforming pathology from manual analysis to intelligent data-driven diagnostics in our core lab. This slide shows 6-plex immunomarker panel on human tonsil tissue that I have worked on Phenocycler Fusion. As we all know, tonsil tissue serves as an ideal model for validating multiplex spatial imaging because of its well-organized tumor microenvironments, including B cell follicles, germinal centers, and T cell zones. Using the cyclical imaging workflow of the Phenocycler Fusion platform, these 6 markers can be visualized within the same tissue section, enabling high-resolution spatial mapping of proliferating immune populations and their organizations within the tonsillar architecture. This multiplex approach highlights how spatial biology technologies can reveal cellular distribution, activation states, and interaction in complex lymphoid tissues. Why Visiopharm? Visiopharm is a flexible platform. It allows development of custom applications, or you can also use a pre-built solutions that are already provided with the software. It reduces reliance on time-consuming manual annotations required by other software. It is very user-friendly. No coding is required. This makes it accessible to a wide range of users. It enhances the reliability and consistency of data. Last but not least, they have excellent customer support. They're very responsive and they're very effective in assisting you. I used the co-occurrence matrix workflow in Visiopharm to analyze spatial relationship between different phenotyped cell populations in multiplex tissue images. After performing cell segmentation and phenotyping, the workflow calculates pairwise proximity between cell types within a defined radius. This generates a co-occurrence matrix that highlights enrichment or avoidance of specific cell-cell interactions. This analysis also helps characterize cellular neighborhoods and spatial organization of tumor microenvironment. Case study 1, glioblastoma. An immunophenotyping panel was applied to glioblastoma tissue using Phenocycler Fusion platform to characterize the spatial immune landscape of tumor microenvironment. The panel includes CD4, CD8 to identify helper and cytotoxic T cell population, CD20 for B cells, FOXP3 for regulatory T cells, and HLA to access antigen presentation activity. PanCK was incorporated to delineate epithelia tumor cell regions and provide structural context for immune cell localization. This multiplex spatial profiling enabled high-resolution mapping of immune cell distribution and their interactions within glioblastoma tissue. The resulting spatial data was analyzed using Phenoplex workflow to perform co-occurrence and neighborhood analysis, enabling quantitative assessment of spatial relationship and cellular interaction within immune and tumor compartments of glioblastoma tissue. Localization of CD4 and CD8 T cells was assessed relative to pan-CK tumor regions to evaluate immune cell distribution within and around tumor compartments. This spatial mapping enabled the identification of T cell infiltration patterns in relation to tumor cell areas and provided insights into tumor immune interactions within the glioblastoma microenvironment. Case study 2, serial section of fetal tissue. Multiplex imaging was performed on serial section of fetal tissue using panel consisting of CD4, CD8, CD20, HLA, FOXP3, and PAM-CK. The multiplex approach allowed comprehensive assessment of immune distribution and microenvironmental organization within the serial section. Using the Phenoplex workflow, multiplex imaging data were processed to perform quantitative spatial analysis of tissue sections. The pipeline enabled measurement of tumor cell density by calculating the number of cells per unit area and estimation of tumor volume across multiple serial sections. In addition, a biomarker staining analysis quantified the proportion of cell expressing the marker of interest. This integrated approach allowed robust cell segmentation, phenotyping, and quantitative characterization of tumor architecture and biomarker distribution. A Disney plot was generated to visualize high-dimensional single-cell biomarker expression data, enabling unsupervised clustering and identification of distinct cellular phenotypes within the analyzed tissue section. Case study 3, breakthrough cancer. Multiplex imaging was performed on fimbriae infundibulum tissue to investigate molecular and cellular signals associated with early cancer-related changes. High-resolution spatial imaging enabled detailed visualization of epithelial structures and surrounding microenvironment across the tissue section. A magnified region highlights the outer epithelia of infundibulum, where biomarker expression patterns were examined to identify potential early tumor-associated signals. This spatial approach allows precise localization of cellular phenotypes within the complex architecture of the tissue. The Phenoplex workflow leverages multiplex imaging to quantitatively assess the spatial landscape of tumor microenvironment. By evaluating the localization sense intensity and co-expression of specific DNA biomarkers, we can move beyond simple protein detection to functional understanding of cellular signaling. This high-resolution mapping is vital for identifying distinct cellular phenotypes and predicting the therapeutic response in precision oncology. Using the Phenoplex workflow, histogram-based data exploration was performed to evaluate the distribution of biomarker intensities in single-cell level. These histograms represent quantitative measurements of biomarker expressions across the analyzed cell population. The plots facilitate assessment of co-localization patterns and expression thresholds for multiple markers. This analysis supports objective gating and identification of cellular phenotypes within multiplex imaging dataset. This slide represents practical workflow for drug target discovery. With its ability to capture the localization, distribution, and quantity of proteins at a subcellular level, spatial proteomics has continued to play a pivotal role in cancer research, uncovering new insights into a cell biological processes underlying the disease. So the first step is to acquire and QC spatial proteomics data, followed by performing robust segmentation and cell type annotations, and constructing spatial graphs, train spatial models to predict outcome and perturbation response, and prioritize candidate targets by spatial specificity, druggability, and cross-cohort validation. Unlocking the potential of antibody-drug conjugate biomarkers is a critical step in enhancing the efficacy, safety, and precision of ADC therapies in oncology and beyond. Antibody-drug conjugates is typically composed of monoclonal antibodies covalently attached to a cytotoxic drug via a chemical linker. It combines both the advantage of high specific targeting ability and highly potent killing effect to achieve accurate and efficient elimination of cancer cells, which has become one of the hotspots for the research and development of anti-cancer drugs. So in our lab, I have performed multiple tissue spatial biology experiments on multiple tissues, including TMAs, esophageal, ovarian, gastric cancer tissues, mouse tongue, some PDX models, and these studies will lead to a development of antibody drug conjugates in near future. I would like to present a case study that underscores the importance of investigating the biological basis of pediatric malignancies. A physician from Boston Children's Hospital provided an autopsy sample for a 9-year-old patient who succumbed to lung cancer. Notably, there was no known family history of the disease. The primary objective was to elucidate the underlying biological mechanism that may have contributed to the development of this malignancy. This investigation was also driven by the parents' desire to better understand the cause of their child's illness. We conducted multiplex immunofluorescence and transcriptomic analysis on autopsy specimens. The biomarker panel was selected by the collaborating physician based on his clinical hypothesis regarding the underlying etiology of the child's disease. The MIF results were consistent with his proposed biological mechanism. To further validate these observations, I applied quantitative and image analysis using Visiopharm, which enabled objective measurement of the spatial and cellular patterns initially identified through qualitative assessment of MIF data. This case highlights that advanced analytical tools such as Visiopharm not only generates high-value biological data for research applications, but can also play a critical role in addressing clinically relevant questions, particularly in complex and unexplained disease presentations. Using the Phenoplex workflow in Visiopharm, I have analyzed multiple tissues, sample types like small bowel tissue, TMA breast cancer samples, and patient-derived organoids. The workflow enabled high-resolution cell segmentation, marker-based phenotyping, and spatial analysis across these distinct tissue architectures. By applying consistent image analysis pipelines, I have been able to quantify cellular populations and evaluate spatial relationship within each sample types. This approach demonstrates the flexibility of Visiopharm platform to handle diverse biological specimens while generating reproducible quantitative data from multiplex imaging dataset. Now, what are the challenges? The integration of AI into clinical oncology presents a complex landscape of hurdles, primarily categorized into technical, biological, and clinical domains. Key technical issues like batch effects and segmentation errors must be addressed alongside biological complexities such as tumor heterogeneity, which can obscure AI interpretability. Ultimately, overcoming these pitfalls, including data privacy barriers and reproducibility concerns, is essential for the scalable and reliable implementation of these tools in patient care. The biological category is particularly critical because it represents the ground truth that AI models often struggle to generalize. Unlike digital data, biological systems are dynamic and inconsistent. So certain issues could be: one, sampling bias. Digital pathology often relies on specific tissue biopsies that may not represent the entire organ or disease state. Second, tumor heterogeneity. A single tumor can contain multiple cell populations with distinct genetic profiles. AI models can train on single snapshot but fail to account for its internal diversity, leading to inaccurate prognostic predictions. Third, low interpretability of AI features. While deep learning can identify patterns, these features often lack a direct biological equivalent that a pathologist can verify, creating a black box effect. Advances in spatial proteomics atlases are significantly enhancing our understanding of cellular diversity and tissue architecture by enabling precise mapping of protein expression within complex tissue environment. When integrated with artificial intelligence, spatial proteomics provides powerful, spatially resolved insights into biological systems, such as tumor microenvironment, allowing for deeper mechanistic understanding of disease processes. These computational approaches improve the identification of clinically relevant targets and refine the accuracy of drug discovery efforts in oncology. As the demand for innovative cancer therapies continues to grow, the combination of AI and spatial proteomics is accelerating the development pipeline from early discovery to clinical translation, positioning these technologies as key drivers of the next generation of more precise and effective cancer treatment. I want to sincerely thank our incredible team for their dedication and expertise, and to everyone who joined us today. I truly appreciate your time and engagement. I look forward to continuing the conversation and advancing this work together. Thank you.
Thank you very much, Namrata. That was really cool. You're really working with different modalities here. That's fantastic.
Yeah, thank you.
Okay, so we already have the questions coming in, so we're just taking them one by one. So Matt says, thank you for the talk. In regard to ACDs, can you add a visual marker to ADCs to enable you to track and see exactly what cells are actually targeted and to what extent?
Yes, the answer is yes, because the ADCs that we are working on, we are looking for specific targets that we want to, like specific gene and specific marker that is associated with that gene, and how much they are going, it is going to express. So Phenocycler Fusion is is the platform that I have been using in my lab for these kind of studies. So all these biomarkers, the biomarkers are barcoded, and they have a very specific localization on where they are going to, are they going to be membranous or or cytoplasmic or nuclear. So it's very specific, and it's been, and after doing the quantitative analysis, we have found out that these biomarkers are able to detect the exactly where it has to be located, and we can work around that area to find out how much and the interaction is there, or what kind of neighborhood, like we can also do neighborhood analysis to know what cells are involved. So as of now, like, because this platform is very new. It used to be CODEX, but now it is Phenocycler Fusion. So they have done a lot of modifications in that. So as of now, we are getting a very good results, and I'm not able to share the biomarkers that I have used because it's a core lab, and but we have been getting excellent results, and this is my fourth project that I'm working on related to ADCs, and customers are very happy about that.
Okay, that sounds very interesting. Do we have more questions coming in? Yes, there's another one. How has Visiopharm's Phenoplex boosted your analysis capabilities beyond the standard multiplex workflow they offer? How would this workflow compare to a spatial transcriptomics approach to the TME?
Yeah, Visiopharm has what Visiopharm has done. It does the like accurate detection and classification of cells that are involved in the particular TME or a particular area that you want to study, and it also quantifies the protein expression. So it also tells you how much or how many cells are involved. It tells you more about like what are the two or more biomarkers they're co-localizing, the co-occurrence, and it tells you which, like the Tisney plot that I mentioned in my presentation, that it tells you where exactly in the cell it is located and how much. So I'm very happy with the quantification part of Visiopharm because it gives a true idea of how many cells are involved. It's not only visual, but it's also quantitative, and it also integrates a lot of multiplex imaging data. Like you can do IHC, IF, all kind of cells, and you can do the if it can be frozen cells, it can be FFPE cells, and also TME. So TMA, tumor microarray. So it can, we can use multiple different multiple tissue types, and it can integrate multiplex imaging data in the software, and it is very scalable, and it's very reproducible, the results. And spatial transcriptomics. So what in our lab we have geomics where we do transcriptomics. So in my opinion, it depends upon what you want to do. Like some people want to do the proteomics part, some want to know the transcriptomics. So mainly I'm working on transcriptomics, and Visiopharm provides data for both transcriptomics and proteomics. So I have used the proteomics, so I can speak about that. So transcriptomics, I'm sure the people who are using Visiopharm for to approach, and the TME that they want to study and quantify, they're having an excellent results too.
Okay. Yes, Michael, thank you for your response. Mats has another question. In regards to the TME, do you consider tertiary lymphoid structures as part of the TME or a separate environment? If separate, how do you relate them to the TME? If part of it, when multiple TLS exists, are they multiple separate sub-environments or one sub-environment?
Yeah, so let me go over the question. Do you consider tertiary lymphoid structures as a part of TME? Yes, I do. And if separate, do you have a little TME? If it's part of the multiple TLS that exist in multiple separate sub-environments. See, our target is to know. Sometimes, the tumor microenvironment is the main issue, where it is. Sometimes they prohibit the drugs or any biomarkers, like all the antibodies, to enter in that in a closed area. So what we are doing, our aim is to target the TMEs and use those biomarkers that are able to they are localized in TMEs, like that can go in the TME area, and then do the quantitative analysis to find out how many cells are affected. But what you're doing when you're doing FFPE or fresh frozen samples that you are using for your studies, so when you're applying a biomarker, you're using it through whole slide, not only TME, or not only a specific structure that you want to study. So based on the biomarkers that you are using, based on what you are targeting, you can we can do the spatial proteomics, we can run the samples, and the results that we get, we can use a platform like Visiopharm to quantify. It doesn't matter if you want to study just the TME, or you want to. So it depends upon what you want to know. You are some of our customers. They have brought a mouse tongue PDX, like PDX model, where they have a human tumor on the mouse tongue, like an oral cancer. So they have a specific requirement. So based on your requirement, based on what biomarkers you want to use, based on what targets you want to study, and you can do anything and everything with these high-plex platform, and then you can do the quantification with Visiopharm. So as of now, I have done small bowel cancers, a lot of breast cancers, studies ovarian cancer, and depending upon what kind of cell we were interested in, like me and the collaborators, we have had a very successful results based on these two platforms, like the studies based on these two platforms.
Okay, sounds good. So we have another question from Catherine. Can you comment on how your existing analysis workflow enables discussions with the biology project teams? What excites the team most?
I've been working on I have started working on the pathology, the histology part, when we did not have the plus chart slides that easily accessible. It was not very cheap. We used to put the egg albumin to stick the slide, the tissue on the slide. So so from there, now when it goes to spatial, like a high-plex platform, we are so much excited to see how specific these biomarkers are working, and how the targeting this, like if you have specific gene in mind, if you have specific clone in mind. So what we do is sometimes our customers, they bring a problem, and then we work together to find out what gene would work. And some of our vendors from the antibodies, sometimes we incorporate them also to find a good clone that would work, and we discuss everything together, and then we start working on that, and then we optimize those biomarkers on this platform. So it's a longer process because you have to optimize. You cannot go from like, sometimes it can be disappointments. Like one of the projects that I'm working on Broad, I have optimized 57 biomarkers for them. So sometimes even 55 work and 2 doesn't work, they get disappointed. Oh, we wanted to do this for our work, but this is what we have to understand, that it's not, because we don't regulate tissue, we cannot change tissue, so it is what it is. So it is interesting to see how, suppose I'm thinking CD4 and CD8, both of them are membrane marker, and they should be I'm just giving an example. So we think that both should localize in the membrane, and it should work perfectly, but sometimes one works, other doesn't. So it's fun to see how these biomarkers work on the same environment and same tissue, and they don't work the way it should be working. So it's a good effort from all of us, and getting a new insight from something that we know that it's going to behave this way, and it doesn't behave. So or it behaves better than what we have thought. This is very exciting for our team.
Okay, that sounds good. So do we have more questions coming up? And I think we can close for the day, and just want to put your attention to the next talk. Please sign up for Shur Asano in about 4 weeks, end of April. And then to Namrata, thank you very much for this excellent talk and for all your insights. It's really exciting to see how all the technologies are being combined to really find new insights in the tissue.
Thank you so much. Thank you so much for having me. It's wonderful to work as a team, and it's beautiful to be learning from each other and helping others learn, and me also learning from others. Yeah.
Yes, yes.
Thank you.
Very nice. Thanks a lot, and thanks everybody for joining, and hopefully see you in 4 weeks. And until then, all the best.
Thank you.
And bye-bye.